Hi friends, welcome to our channel. Today I will discuss about the topic of SiRNA. First we see what is SiRNA. SiRNA is nothing but a small interfering RNA or short interfering RNA or slicing RNA. SiRNA is a class of double stranded RNA molecule. SiRNA is a double stranded RNA molecule which length has 20 to 25 base pairs which operating within the RNA interference pathway. This is a short note about siRNA. Next we see the structure of siRNA. Naturally occurring siRNA have a well-defined structure, that is a short double-stranded RNA with phosphorylated 5' end and hydroxylated 3' end with two overhanging nucleotides. In this structure, there are 3' and 5' ends are present. In upper portion is called as guide strand. In lower portion is called passenger strand. The guide strand is nothing but a antisense strand. The passenger strand is nothing but sense strand. So the sense and antisense, the two strands are present in the siRNA. In overhanging portion, there are nucleotides present. In overhanging, there are two nucleotides present, so is called as dinucleotide overhangs. This is the structure of siRNA. Next, we see the synthesis of siRNA. Before that, we see what are the guidelines for siRNA synthesis. First one, siRNA targeted sequence is usually 21 in length, already known 21 in length. And second one, to avoid regions within 50 to 100 bb of the start codon and the termination codon, to avoid intron exchange, to avoid repeats and low complex sequences, to avoid single nucleotide polymorphisms. These 6 things are the guidelines, the basic guidelines for synthesis of siRNA. Next we see what are the methods to be used for synthesis of siRNA. First one is chemical synthesis. Second one, in vitro transcription. Third one, RNase or dicer digestion of long double-stranded RNA. First we see the chemical synthesis. In chemical synthesis is a most expensive method. Chemical synthesis is a most expensive method is to produce high quality of chemically synthesized siRNA on custom basis to yield the large amount of high purity siRNAs. This is the synthesis of chemical synthesis of siRNA. The chemical synthesis is best for the studies that require a large amount of defined siRNA sequence. The chemical synthesis is best for to require high amount of defined siRNA sequence, not suitable for screening of siRNA sequence and long-term studies, not suitable for screening methods, suitable for large amount of defined siRNA sequence produced. This is the chemical synthesis. Next we see the mechanism of siRNA. In this mechanism, there are 6 steps are present. First one is dicer cleavage of dsRNA, such that dicer cleavage of double-stranded RNA. Dicer is nothing but endoribonuclease, which used to cut the double-stranded RNA. The second step is population of siRNAs duplex formation. Third step is single-stranded incorporation into RISC. The RISC is nothing but RNA induced slicing complex. RISC is nothing but RNA inducing slicing complex. The fourth step is strand unwinding and separation. Fifth step, targeting specific mRNA molecule. Then last step is slicer mediated cleavage. RNA degraded by exonuclease. The 6 steps involved in the mechanism of siRNA. Next we see the mechanism of siRNA in detail. First thing, the long double-stranded RNA is cleaved by endoribonuclease called dicer. The dicer cuts the long dsRNA to form short interfering RNA or siRNA. It enables the molecules to form the RNA induced slicing complex such that RISC. Once the siRNA is a part of RISC complex, the siRNA is unwound to form single stranded siRNA. The strand is less stable due to base pair at 5' end. Once single stranded siRNA binds to the target mRNA, it induces mRNA cleavage. mRNA is now cut and recognized abnormally by the cell. This causes degradation of mRNA and in turn no translation of mRNA to amino acid and then proteins, thus slicing the gene that encodes that mRNA. This is the mechanism of siRNA. Next we see the importance of siRNA. The first thing is siRNA are widely used to access the individual contribution of genes. Second important is and to an assortment of cellular phenotypes including cytokines, apoptosis, insulin signaling and cell differentiation. Next, siRNA screens have been used to identify the novel pathways such that have significant impact in validating targets for a number of cellular processes and disease including cancer, HIV and hepatitis. Finally, in vivo RNA has been used for target validation studies in animal study disease model and has the potential to be used for therapeutic purposes where disease causing genes are selectively targeted and suppressed. Example, cancer. The disease causing genes are selectively targeted and suppressed. Next, when a mammal cell is faced with a double-stranded RNA such as a siRNA, it may mistake it as a viral byproduct and initiate an immune response. In addition, the introduction of an siRNA may cause unintended off-targeting where other non-threatening protein may also be attacked and knocked out. This is the importance of siRNA. Next we see the chemically modified siRNA. First point, enhanced activity. The chemically modified siRNA has enhanced the activity. It increased the serum stability and less off targets. Third point, it decreased the immunological activation. Then many diseases can potentially be treated by inhibiting gene expression. There, the design of synthetic siRNA for therapeutic uses has become a popular objective of many pharmaceutical companies. This is the importance and significance of chemically modifying siRNAs. Next, finally, we see the application of siRNA. First one, testing hypothesis of gene function. First, know the gene function. Second one, target validation. Then pathway analysis. Then functional screening. And finally, siRNA as therapeutics. This is the application of siRNA. Today we discussed the what is siRNA, then structure of siRNA, then synthesis of RNA, then mechanism of siRNA, then what are the importance of siRNAs, then chemically modified siRNA, such that significance of chemically modifying siRNA compared to commercial normal siRNAs, then finally in the application of siRNA. Thank you.